We characterize the in vivo mechanisms of aggregate and organelle extrusion from neurons via a process called exopher. Poorly understood biology that's likely relevant to neurodegenerative disease pathology spread.
These approaches are necessary for achieving reproducible scoring of extruded neuronal exophers and demonstrate a platform for the mechanistic dissection of cellular trash elimination via neuronal transfer to neighboring cells.
Defining the in vivo mechanisms of how neurons eject aggregates may actually influence the design of novel strategies to aid in the treatment of human neurodegenerative conditions.
For basal conditions, house C. elegans at the same biological age at a constant temperature of 20 degrees Celsius. For live imaging of the intracellular dynamics and characteristics of exopher, place up to 20 animals onto an agarose pad on a glass microscope slide, and immobilize the animals. Carefully place a cover slip over the paralyzed worms, and place the slide onto the stage of a confocal fluorescence microscope. Use a 10 to 40x objective to identify the desired Z-plane in bright field, taking notes of the positioning of the worm, the head-tail orientation, and the vulva location, which are landmarks for later neuronal and exopher identification. Staying in the same Z-plane, switch to wide-field fluorescence viewing at 10 to 40x for the chosen cytosolic reporter, and scroll within the Z-axis to observe the depth of the animal and the fluorescence expression in the focal plane. The head will have a fluorescent nerve ring, and the more pointed tail will contain 1 to 2 visible posterior lateral touch neurons.